Good morning, everyone. Um, Sorry, I just woke up a bit late. I was sleeping late night, working late night last night, so that's why. Yeah. So, till where we did last time, I think we have to start from DNA replication, right? Today we have discussed about central dogma and everything was done. We have to start with DNA replication, isn't it? Please confirm me, students, that we have to start from DNA replication today. Okay, fine. There are only four concurrent viewers I could see. There are a lot of other students need to join actually. Let's wait for 1 minute more, then we start. Okay, from tRNA, then we start. Okay, so yesterday we were discussing about, we have shown that very nice animation about transcription and translation. How that central dogma of life working in very synchronization way in order to make our body happy and healthy. So in that sense, let's discuss now the transfer RNA. So it's a small RNA chain of about 80 nucleotides that transfers the specific amino acids to growing polypeptide chains at the ribosomal site of protein synthesis during the translation. So it has this mRNA, it has three codons, that is GCC codon, and there's anticodons, that is CGG on the tRNA at the top. So it has amino acid attachments and an anticodon regions for the codon recognitions here. And also it has a region for attaching the amino acids on the top, which could be attached with the help of ester bonds, right? So this sites binds to specific sequence on the messenger RNA through the hydrogen bonds. So here they are attaching via the hydrogen bonds. On the top, the tRNA is attaching to the amino acids via the ester bond. Then there's ribosomal RNAs. There's mainly two ribosomal RNAs that are present, 70S and 80S. 70S is made from 50S and 30, and whereas 80 is made out of 60X and 40S. That's and then they goes into the cytoplasm, and with the help of tRNA, they help in the production of our corresponding polypeptide chain of our proteins. So main difference between DNA and RNA so far, this is ribonucleic acid, this is deoxyribonucleic acid. It has ribose, it has deoxyribose sugar. It's a single stranded, it's a double stranded mostly. On the second carbon, the hydroxyl group is removed from the RNA, and the DNA it is still sorry, in DNA the hydroxyl group is removed. Here it doesn't. Instead of thymine, we have uracil, and here is thymine. That's the main difference. And proteins that are also known as polypeptides are made up of amino acids arranged in a linear chain folded into a globular form. And the sequence of amino acids in a protein is defined by the sequence of a gene, which is encoded in the genetic code. So genetic code specifies 20 standard amino acids. So here we have the polypeptide chain of amino acids, right? Each cysteine with another cysteine, they are making this disulfide bridges. They are making these disulfide bridges, and then they make a quaternary structure. They got folded, in which the protein is active, and this could be used in our body then. So it could be present in peanuts, pulses, red kidney beans, fish, um, chickpea, red meat, eggs, chicken breast, and all these things they are present. So at the end, I would say what we have studied so far, the DNA, RNA proteins, they are the major, like basic players who are playing role in this molecular biology or biochemistry. So DNA could replicate, self-replicate, where it could then go for transcription, leading to production of RNA, and from RNA, protein is produced with the help of a wire translation process, and this whole process is known as central dogma. Now comes the another part, that is the DNA replication. So as we know, in our body as a human being, DNA replication is the basis for biological inheritance. It is a fundamental process occurring in a living organisms to copy their DNA. So in the process of replication, each strand of the original double-stranded DNA molecule serve as a template for the reproduction of the complementary strand. So two identical DNA molecules have been produced from the single double-stranded DNA molecules. So this is a semi-conservative in nature. So as we know, so if there are two strands, this is like father, parents, parents DNA. So when it's gonna differentiate into two parts, so half of the genes will come from this one and half from the other. So 50-50% it will go forward. That's why it's also known as semi-conservative nature, right? So let's see how this process is done, this DNA replications. Let's check the main enzymes that are playing role in a nutshell within 2 minutes view of our lecture here. So here we can see this is 5' end of our DNA. 5' end of DNA, which is going to this 3' end here. 5' end, 3' end. On the other hand, this is a 3' end, making a 5' end here. Okay, so what is happening is, first thing is topoisomerase. This enzyme comes and it's unwind the double helix structure. What other double helix structure was there? It's just opened up, and then the bonding between the two nitrogenous base pairs, the hydrogen bonds, they will be broken with the help of second enzyme, that is helicase enzyme, right? After this bonds are broken, in order to have stabilized nucleotides over the template of these strands, our third thing is single stranded binding proteins. They will come into the role, and they help in the stabilization of our nucleotides, right? So in that sense, when the things got open, there will be two strands will be produced. One will be the lagging strand and leading strand. Okay, the leading strand, what will happen, it tend to move from 5 prime to 3 prime end. So whatever the DNA replication will happen, it will always happen from 5 prime to 3 prime direction. So it will start with the help of enzyme now. So we have discussed topoisomerase, helicase, are the single stranded binding protein so far. So 3 enzymes we have discussed. Then the fourth enzyme will play into the role, that is DNA polymerase. So this will come and start producing the corresponding DNA nucleotides, and it start to fold again, right? It start to fold again. On the other side, in the lagging strand, what it will happen? So this will be your 5 prime end, yeah, and that is your 3 prime end. So the DNA replication will happen in the opposite direction of the leading strand. And while it is happening, there will be some nicks here. This is called nicks, some gaps. So these gaps make fragments of our DNA, fragments produced by the DNA polymerase. These fragments are called as Okazaki fragments, discovered by the Japanese scientist. So this could be recovered with the help of DNA ligase. So DNA ligase will join them together in order to make the things. And initially, in order to start the reaction, we will have RNA primer and DNA primase will be there in order to produce that. So whenever you have a question about DNA replication, just unwind it with the help of enzyme, add helicase, then single stranded binding proteins, then DNA polymerase, then divide into leading and lagging strand, and then always make your replication from 5 prime to 3 prime end, and then join all the major enzymes that are required into it, right? So in a cell, DNA replications begins at specific locations in the genome called origins. An unwinding of DNA at the origin and synthesis of new strands forms a replication fork. So we can see uh, like a cutlery that we had in a house. So it's become like a replication fork with two forks here. Yeah. In addition, DNA polymerase, the enzyme that synthesizes the new DNA by adding nucleotides matched to the template strands. A number of proteins are associated with the fork and assist in the initiation and continuation of the DNA synthesis. Then cellular proofreading that ensures the neat, perfect fidelity for the DNA replication is done. So in this way, the whole DNA replication works. Now transcription, though I will not go in much details here, just because we have discussed them in yesterday. So is the process of creating an equivalent RNA copy of sequence of DNA. Yeah, that's the first point. I hope my yeah, okay. So everyone is there. Yeah, it's a process of creating an equivalent amount of RNA copy from the sequence of DNA sequence. So whatever the amount of DNA is there, it will produce the same amount of RNA copy. So in this, the first step leading to the gene expression. So DNA is producing your RNA. This is called transcription. And if RNA is producing DNA, then it's called as reverse transcription. And during this transcription, DNA sequence is read by the RNA polymerase, which produces complementary antiparallel strands. So RNA polymerase is the key enzyme which is helping in the production of your RNA. And also in transcription, RNA complement includes uracil instead of thymine. So whatever the whenever the A is there on the top, instead of T on the mRNA, it will produce U. So during this, the stretch of DNA that has to be go from a DNA part into the RNA molecule, it should encode, at least one gene has to encode. So if the gene transcribes the proteins, the results of transcription is known as messenger RNA. So why is it called messenger RNA? Basically, it's act as a messenger, like a postman who is taking the message from one point to another point. So he's reading the DNA message, what it is, in the form of messenger RNA. Then it's giving it to the outside the nucleus to the ribosomes and tRNA, and then they are reading it and making the form of protein. So in between, it's acting as a mediator, nothing more than that. So mRNA will be used to create the proteins for the process of translation in this case, and DNA transcription units encoding for the protein and regulatory sequence that directs and regulates the synthesis of that protein. So here we can see DNA is read from the direction of 3' to 5' direction. So 3' to 5' direction is there. But your complementary RNA production, it will always happen from 5' to 3' direction. Right, and the RNA polymerase will help us to produce your corresponding mRNA. And only one RNA will be produced in this case, because one of the template is being used for the same, not two templates will be used. The other DNA, the strands that we'll be using will be called as coding strand here. So we will have at the end this single stranded RNA, mRNA, transcribed with the help of RNA polymerase. With the help, also it will have hairpin loop here, we can see very nicely, and yeah, then single stranded again. So 5' to 3' end. And also during this process, there is called RNA synthesis and processing. It's these things that I'm describing you. They are actually very quite long lectures, but I'm just trying to give you in summary about all these at the moment. So we have your chromosomal DNA, which will have your exon 1, exon 2, exon 3. Yeah? This is the whole gene is there. And the intron parts, these are the introns part. So we can remove them. So these are non-coding regions, these are the coding regions. These are the coding regions, and these are the introns are the non-coding region. Right. Then after the transcription, what will happen? The external introns will be removed. These external, they will be removed. Still internal introns will be there. So this will be also removed during the RNA splicing. Then at the end you have your messenger RNA and RNA synthesis. Then at the end you will have all exons 1, 2, 3 to be present there. Right. Then reverse transcribing viruses, which replicate the genomes by reverse transcribing the DNA copies from their RNA, and these copies then enter to new RNA. So what happen is in the viruses, the HIV RNA, it will produce HIV DNA with the help of reverse transcriptase enzyme. So this is the something reverse cycle is happening in the case of RNAs or viruses. So again, this is the in general your in the central dogma. So your DNA could replicate, produce RNA, and it go for the protein. But then there are some special cases, RNA could produce DNA with the help of reverse transcriptase, and RNA could also self-replicate itself. So all these things are possible within the case of central dogma of life. Always remember, this is a case of general and special, and remember, it will never happen that DNA is producing proteins. Yeah, this is not possible. Or protein is producing RNA. This is never possible. Or protein is producing DNA. This is never possible. So these three scenarios within the central dogma of life, they are never ever possible. Okay. Now comes translation, which is the first stage of protein biosynthesis. So in this, your mRNA bound to the ribosomes and they start to produce long chain of uh, amino acids. Then they get folded together in order to form the active proteins. And this happens in the cytoplasm where your large and small subunit of ribosomes are located and they bind to the mRNA. So here we can see our messenger RNA 5 prime to 3 prime end first. Yeah, just draw if you want. Sometimes it comes for an exam to draw this translation part. Just draw this messenger RNA first with the 3 amino acids as a genetic codes together. Then make this ribosomes smaller and larger subunit. Yeah. Then write down valine, alanine, glycine. These are the 3 amino acids that are present over the messenger RNA. Then what will happen? Your tRNA will come and join to the respective uh, codons of your messenger RNA, and the tRNA will have the corresponding amino acids on top. They will come and read them, and your corresponding polypeptide of amino acids will join it, and it's become a protein chain of amino acids here. And this cycle, it will get removed then, like this tRNA of valine is removed after getting attached, alanine is attached, then the glycine will come, it will attach to it, then phenylalanine will come, it will attach to it. So in this way our long amino acid chain will be produced. So the entire ribosome mRNA complex will bind to the outer membrane of endoplasmic reticulum, and the release of nascent proteins inside the later vesicle transport and secretion outside the cell is done. So in this way. And then this is a very important part, the genetic code of a human. That is, in order to draw this, for what you do is first write down UCAG. Yeah, UCAG here. Then write down on the second base UCAG, right? And then on the right hand side, on third base pair, you write all 4 letters in each box: UCAG, 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 right? And then start writing them. Take one alphabet from here, one alphabet from here, and one from there. So UUU, then UUC, then UUA, and UUG. So like this, you will have around 16 boxes, 4 multiplied by 4, and you draw all these amino acids and start giving them name. You can start giving them name from the start codon here. That is your methionine, which is the start codon AUG. Always remember. And then there is stop codons, that is UAA, UAG, and UGA. Yeah, give them the names. Then start giving names towards the big boxes which has these common names, like first is serine, right? Then leucine, then proline, then threonine, then valine, alanine, arginine, glycine. So in this way most of your boxes will be completed. Then start giving others name one by one: phenylalanine, leucine, and then this is all leucine again. Then check, this is tyrosine, and these two are cysteines, so on, histidine, glutamine, asparagine. So these names should be remembered. If in exam it's come, you can draw this diagram and you should remember all these names. What is GAU, what is GAC? It's really helpful actually from subjective perspective, for your future exams perspective, or for general knowledge also. So this genetic code table might be very helpful for your various perspective. Okay. Then what is genome? Right? What is genome? Um, so we are done with the first part of our subjective with the whole studies about DNA. We have discussed about RNA. We have discussed various types of DNA. Then we have discussed various types of RNA. Then we have discussed the difference between DNA and RNA. Then we went for transcription process. Then we have discussed about translation process just now in details, and we have discussed about the genetic code. Now I'm gonna take to the next chapter of this basics of biochemistry and molecular biology, that is genome. What is genome is? So genome is entirely an organism hereditary information. It is coded either in the DNA or for many types of viruses. So genome includes, that is, genes and non-coding sequence. So at the moment in our genome, human genome, it has 98% is non-coding. We don't know the reason why is 98% is there, but only the 2% of our whole genome of our body, it's coding for protein. They are the one which are coding for the protein, right? And within the genomics, there is a field called comparative genomics, which compare the genome sizes of different organisms. So it's very nice table explaining um, all the major models to study diseases or to discover some good medicines or to discover some healthy products. So there are 8 models written here, right? So Homo sapiens, Mosmosulus, right? Drosophila melongaster, Arabidopsis thaliana, Chinorhabditis elegans, Saccharomyces cerevisiae, Escherichia, and Haemophilus influenza. So all these things, they are the basic models that we use, and you can check that the humans has around 3.2 billion base pairs of the genes, and here it has 46 chromosomes. And that's why we choose mouse as a model for experiments, because they are almost similar, 2.6 billion base pairs and 40 chromosomes are there. Yeah. If somebody is interested for the study of genetics, then they go for fruit fly model. For in genetic studies, they are the best model because of the presence of 137 millions base pair of genes, but it has 8 chromosomes only. Then in the case of plants, if somebody is interested, this is the best model, Arabidopsis thaliana, which has 100 million base pairs and has around 10 chromosomes. And the yeast is also one of the best model because it has around 12.1 million base pairs and it has almost nearby 32 chromosomes. Yeah. So for the human studies, if something is found within the mouse or within the yeast, it's readily acceptable in the world. Things could be tested in the humans afterwards. Then in the case of prokaryotes, they have only one chromosome, yeah, with 4.6 and 1.8 million base pairs of your genes, respectively, in E. coli and influenza, H influenza. So this table speaks a lot for himself in order to compare between different genome sizes and speaking its various key respectful to that. Okay, so now comes the various tools that they are being used in the case of molecular biology and biochemistry. The first tool that we used is gel electrophoresis. Is gel electrophoresis. The basic principle is that DNA, RNA, proteins, can all be separated by means of an electric field. So you have a negative electrode here and you have a positive electrode here. And tell me why our DNA is negative electrode, negatively charged. So DNA further will be passed out through the gel, and it will separate it because of its sizes. So the big molecules will be up, the middle molecules will be in the center, and the small molecules will be even at the bottom. So please explain that why this in the gel things are like this. So why, I mean, why your DNA is negatively charged? Can anyone tell me in the class? Comment in the box. Anyone else? Anyone else, students? Basically, it's because of the phosphate group. That's right. That's why it moves from your negative side to positive side, and we can separate our DNA. So DNA could be separated on the agarose gel, and the proteins could be separated via the STS page, that is sodium dodecyl polyacromate gel electrophoresis. Um, and also we can use 2D gel electrophoresis. So these two things are possible in this case, and we can separate our protein. Then there is a polymerase chain reaction, which is an extremely versatile technique in order to copy millions copy of our DNA. So PCR allows a single DNA sequence to be copied or altered in the predetermined way. So there are many ways in order to produce it via amplifications of our RNA in order to understand the particular DNA or RNA. So what happen is, so there are three steps are happening, that is denaturation, annealing, and elongation. Yeah. So during this denaturation, what is happening is your DNA are separated. Your double helix structures, which is joined together, they are separated, and then the primers are being attached here in this annealing step to the corresponding DNA. Then in the elongation process, they start to produce the corresponding sequence afterwards. So denaturation, uh, annealing, and elongation. So we want this gene to be grow. So our template DNA, after the first cycle of DAE, it will produce two different copies, like 4 copies. Then in the third cycle, it will produce 8, 16, 32. By the 35th cycle, we will have around 68 billion copies. So it exponentially grows actually. Your PCR, the DNA grows exponentially. The primers that we used in this of nucleic acid serve as a starting point for DNA synthesis, and they are mostly around 80 nucleotide ends. And replications always starts from 3' end of the primer and copies the opposite strand. It has tons of millions of applications of PCR. Common application is to understand the specific patterns of gene expression, and the task of DNA sequence can be also be assisted by the PCR, right? And PCR has numerous applications to form traditional processes of DNA cloning, and many more, for genetic mapping, for DNA fingerprinting, in order to identify the parents, parental testing, so many applications. So let's watch one virtual video on the PCR, that how PCR is done.
Before beginning this protocol, students must fill out the worksheet provided on the last page of this document. This will allow students to calculate the appropriate volume of reagents to mix in the creation of a master cocktail. The purpose of a cocktail is to mix all the common reagents in a multi-sample procedure to avoid excess pipetting and potential errors. In this example of Protocol 2A, 4 students are amplifying the actinin 3 gene from all 4 of their DNA samples. This information is filled out on the left-hand side of the worksheet. On the right-hand side, students use the equations to determine the number of tubes needed and the multiplier for reagent volumes. In addition to the 4 DNA samples, a negative control sample is always included. Therefore, 5 tubes are needed for the PCR reaction. In the creation of the cocktail, pipetting error is accounted for by adding another sample. Therefore, the multiplier for this group is 6. Each row of Table 1 represents a common reagent used in all the samples undergoing a PCR reaction. The multiplier is filled out in all rows of the multiplier column of Table 1. Moving across each row, the volume indicated per reaction is multiplied by the multiplier. .5 times 6 equals 75 microliters of red tack added to the cocktail. 11.5 times 6 equals 69 microliters of water added to the cocktail. 0.5 times 6 equals 3 microliters of each of the forward and reverse primer added to the cocktail. Finally, the sum of these volumes should be equal to the product of the final row. 75 plus 69 plus 3 plus 3 equals 150 microliters of total volume in the cocktail. This amount matches 25 times 6.
So understand, student, so what they are doing, we have our sigma tag ready mix, which is required for the reaction. Then there is a water, there is a forward primer, then there is a reverse primer, and then this is the total cocktail volume. So 12.5, 11.5, 0.5, 0.5. So they are preparing this DNA sample for 4, and there is one control and one positive control and one negative control. So total we have to create 6 samples. So that's why we make 6 here. Then we multiply each value 12.5 by 6 and this, 0.5 multiplied by 6, we have 3, and the addition was 150. Then afterwards, the total core volume 25 multiplied by 6 is 150. So that's what they are doing at the moment.
Table 2 is used as a guide for labeling of the PCR samples. As indicated by the equations, this group will be using 5 tubes. The class must collectively number the PCR samples to not confuse samples amongst the groups. In this example, these students have been assigned samples numbered 5 through 9. The sample names are filled out in the second column, and the primers used for each sample are filled out in the last 2 columns. In this example, the forward primer is actinin 3F, and the reverse primer is actinin 3R. The same primers are used for all samples. The common reagents provided are molecular biology water, a forward primer, which you should be sure to check for the marking of PCR to ensure that you are using the appropriate concentration, a reverse primer, and red tack ready mix. The red tack will need to be thawed prior to use, but once thawed, keep the solution on ice to prevent enzyme degradation. Next, prepare the cocktail by using the volumes and reagents indicated on the worksheet. Obtain a new tube and label it as the cocktail. When creating a cocktail, it is best to start with the largest volumes. Though red tack is the largest volume, the solution should be kept on ice as long as possible, so instead the water is added first. When adding each of the reagents, be sure to use the appropriate micropipetter for the volume being added. Next, the primers are added. It is best to pipette small volumes directly into larger volumes to avoid loss of the reagent. Finally, add the red tack and immediately return the stock to ice. Mix the solution and centrifuge all cocktail tubes created by all groups in the class together. Press and hold the short button to do a brief spin.
Okay, that was the part one. I hope you are understanding the major differences between your. Let's start with the second part.
The collected cocktail solution will be split amongst the 5 tubes in this example. PCR tubes are provided in strips of 4, so if you need 5 tubes, use 2 strips of tubes. In this case, to amplify actinin 3, 24 microliters of cocktail solution is added to each of the 5 tubes. Before adding DNA to the PCR reactions, make sure that the tubes are labeled. Make sure all DNA samples are mixed before transferring the solution. One microliter is a very small volume, but you should still be able to see a drop in the tip. Put the tip all the way into the bottom of the tube and pump up and down on the plunger before expelling to the second stop. This helps to ensure that the 1 microliter of DNA has been expelled into the reaction. the tubes tightly and flick to mix the samples. Use the mini microcentrifuge with the 8-tube strip head to collect the solution at the bottom of the tube. It is good practice to always mix and collect solutions before every step, especially if the solutions have been sitting for a while. Turn the thermocycler on with the switch in the back. Place the tubes in the thermocycler by pressing the metal latch and lifting up on the lid. Tubes can be placed in any spot or orientation. Close the lid and make sure the metal latch has clicked into place. Heat cycling programs have already been programmed into the thermocycler, so all you need to do is run them by pressing the Proceed button. Use the horizontal arrows to navigate to the program of interest, in this case ACTN, to amplify the actinin 3 locus, and hit Proceed. The heated lid should be enabled by hitting Proceed to prevent extreme condensation at the top of the tube. The thermocycler will ramp to the initial temperature and begin timing the incubation. It is good practice to make sure the machine reaches this step with no errors. After it has initiated properly, the machine can be left while the reaction proceeds for several hours. Once the run is complete, the cycler can be turned off by simply flipping the switch in the back of the machine.
Okay. So our PCR is over by doing so. So I will share all these things with you now. This one I have shared with you already, the introduction of basics molecular biology and also the structure of DNA. So yeah, all material have been shared with you already. So that's good. So that's it for today, students. You are done with the PCR, and tomorrow we will do with the little bit of blotting techniques, and then we will switch to the more about because initially we want to do with the everything related to the nucleic acids, nucleotides, nucleosides, DNA, RNA, translation, transcription, understand about the basics of them, what is it all about, and then we gradually move towards the amino acids, lipids, carbohydrates, about the basics chemistry behind them, and then go for further uh, lipid metabolism, carbohydrate metabolism, and so on. Yeah, so that's it for today, students. We will see us then tomorrow, yeah, at the same time, at the same place, and continue further. Maybe I will take your classes on Saturdays also, because your syllabus is huge and I might not able to finish in 1 month. So in order to finish your things, we might have classes on Saturdays also. So I will take mainly Sunday off for all of us. Okay. So we see us tomorrow at 7 a.m. in the morning. And then we continue further. So thank you.